that was statistically significant after Bonferroni correction. On this graph, you have the chromosomes along the x-axis, so each of these dots represent a genetic variant that was tested. And on the y-axis, we have the negative log of the p-value. This was our top variant, and it was linked to one other variant, which was in the same gene, the folio-polyglutamate synthase gene. They were not linked to any other variants, just those two linked together, and that's why it's such a sparse peak in the Manhattan plot. So, what is this variant? This variant occurs just outside of exon 2, and the irony is not lost on me that we used exome sequencing to find something that might be likely regulatory. It was not linked to any amino acid changing variants, and it had an allele frequency of 20% in our discovery cohort. Interestingly enough, when you look at the map, you see that this variant is pretty much just shows up in sub-Saharan Africa. And as I mentioned before, most research is done in non-African populations. So all of the warfarin studies that have been done up until this time, all of the studies that have focused on European populations and Asian populations, completely miss this variant because this variant was actually not in any of those populations. So what does this gene actually do? This gene is involved in folate homeostasis. Folate is a vitamin that you've probably heard of before. It adds glutamate moieties onto the folate and helps decide whether the folate is kept inside the cell or outside of the cell. Our variant was associated with a lower warfarin dose. We replicated using all of the available additional African Americans on warfarin that we could find, 373 individuals, which were completely distinct from our discovery cohort, and these people had doses that were normally distributed. We joined together the two cohorts in order to do an analysis where we wanted to make a rough estimation of exactly how much this variant lowered warfarin dose by. And to do this, we used the IWPC equation, which I had mentioned earlier. We found that each copy of this variant lowers warfarin dose by about 6 milligrams a week, which is a clinically significant dose lowering. As I mentioned before, this variant is not a protein code changing variant, and so we were interested in trying to figure out exactly what this variant does to its gene that it's right outside of. We got lymphoblastoid cell lines from the HapMap project. We collected information from cell lines of African descent because African American data was not available. However, African Americans are about 80% West African descent, so it's a good proxy population to use. And we looked to see if the levels of FPGS expression changed with the risk allele. And we found that with every copy of the risk allele, you actually have a statistically significant lowering of the gene expression. So, next question that we were wondering is, what role does warfarin play with folate? What is the interaction between the two, if there is any? And actually, there's literature on that as well. There was a small study using 24 individuals that found that if you increase your folate intake, you actually increase your metabolism of warfarin. Numerous studies have demonstrated that folate is incredibly important for coagulation or clotting status. And additionally, this gene is highly expressed in the liver, which is the site of warfarin metabolism. Additionally, with the recent release of the human proteome map data, we see that the FPGS protein is available, highest level in the human ovary, but the next highest level is human liver. So we have both genetic and protein evidence for the importance of FPGS in the liver, which is, as I mentioned before, the site of warfarin metabolism. Based on this information, we conclude that this variant is a cis-EQTL, meaning that it's regulating the neighboring gene, and it's in folate homeostasis, and it's associated with lower warfarin dose in African Americans. While there has been plenty of biological information tying folate to warfarin, this is presenting the first genetic evidence that the folate homeostasis pathway is tied to warfarin. Based on this body of work, I conclude that we had a non-synonymous population-specific variant in protein S, which was associated with clotting in African Americans. This is one of the first mutations that have been documented in African Americans to affect their clotting risk. We had a regulatory population-specific variant in the folate homeostasis pathway that was involved with influencing the final dose of warfarin. And finally, as I hope you will agree with me after these two stories, population-specific variation is incredibly important for explaining phenotypic variation across different populations. I have a list of my publications because there are so many individuals who have been involved in this work, and I would like to at least put their names up there. The two pieces that I presented today are currently in the process of being published. I have many acknowledgments to give, but I'm not going to stand here and acknowledge for the next 2 days. But I would first like to acknowledge Russ and the Altman Lab and Farm GKB, Carlos and the Bustamante Lab, Terry Klein for all of her advice and support and help, my many, many collaborators, my thesis committee, Dr. Therio, Russ, Carlos, Atul, and Mike, Maura and Lori, the MSTP program and directors for taking me in, the genetics department, and of course you always have to thank the people who give you money, so I'd like to thank my funding. I would also like to thank my academic and life advisor, Dr. Salvatierra, Shar and Zira, all of my friends, many of them who came here today, many of them who are now residents, my parents for having me, my grandparents for having them, my little brother for being who he is. That's him right there. My husband for always being my biggest supporter and who I met actually right outside Nexus Cafe 5 years ago, and who married someone who was just going to get an MD, and when I came and told him that I was going to get a PhD, his response was, well, I knew you were going to do that. So, thank you very much, and I'm very happy to take any of your questions.
So, I'm aware of your opinions on the use of racial classifications in medicine, and they're quite valid. So then,